moves through. It's sort of there's circular and there's longitudinal muscle that go through the that are pervasive all the way throughout the digestive tract. The circular muscles sort of squeeze it like if you had straw, like Play-Doh in a straw, and it was filled. There's a big bunch of Play-Doh in there, and you just squeeze it right in the middle. You kind of squish it down. That would be like what circular muscle could do. Now, if you could act like it was like one of those crinkly, bendy straws or whatever, that you could go like this and pull it back. If you could have that happen at the same time, sort of squish down and contract lengthwise at the same time, what you can get is propulsive movement. Not only do you get mixing together, but you can get smushing it, mixing it up, and moving it in one direction as well, sort of like a snake. So we've got motility, we've got secretion. There's a lot of activity that's going to be happening in terms of hormones being released, hydrochloric acid being released, enzymes being released. So there's a lot of secretion that goes on in the stomach, as well as the duodenum, the beginning of the small intestine. Then we've got actual digestion. And digestion is actually, okay, I don't know if you'll see this again somewhere or not. I don't know if it's like on the study guide, if it might say something like, what is digestion actually from a chemical perspective, something like that. It is hydrolysis. I'm going to discuss what hydrolysis is here in a little bit. It's a chemical reaction that involves water. Okay? It allows us to take large molecules. We've got carbohydrates, proteins, fats. It allows us to take these and use water molecules to help bust them up and pull them apart into smaller pieces. That's what digestion is. We'll take large carbohydrates like starch, bust them down into glucose. Proteins can be busted into amino acids, etc. All right, absorption is then when we take all these things after all this has happened, we take those things that are floating around out there now and pull them back up into the bloodstream. That's absorption. That's the final thing we'll be talking about. Okay? We've got water, vitamins, electrolytes. All that stuff's going to head into the blood and the lymph, and the nutrient molecules as well are going to head into those places. And we're going to look at some of these processes. This is just something that you should already know in terms of what secretion is, what exocrine versus endocrine is, that kind of thing. What is the difference between endocrine and exocrine? And an endocrine, with an endocrine secretion, if you have cells that are endocrine cells that are making a product and they are secreting those cells, secreting that product, where does that product go if it is endocrine? Into the blood. If it is exocrine, it goes into like a lumen or a cavity somewhere within that organ. Okay, like here, if these are some sort of duct that's attached to some kind of hollow tube, some lumen up here, if the products are secreted into here and make their way up through here, this is a exocrine function. Okay, this is an exocrine secretion. Now, endocrine would mean that if these were cells that were releasing products, it would go out into the bloodstream and then travel around. Okay, so that's the difference between the two. All right, now, as far as the motility goes, in terms of the smooth muscle, that's what's going to be contracting. Fortunately, we don't have to think about it, right? It's autonomous, automatically controlled, smooth muscle. We don't have to focus our efforts on moving our food through our guts, as we say here. Okay, it does that. Our body does that for ourselves. Now, there always is going to be tone. We talked about this with the muscle system, a low level of constant contraction that's sort of just keeping some tension there. We've got propulsive and mixing movements. Mixing, what you'll see is sort of like if we had this tube and there was some stuff inside of it, if you just kind of squeeze it back and forth and mix it all up, and then, don't always squeeze in the same spot, squeeze it here, then grab here and squeeze it, and then keep moving it back and forth, you can mix stuff up. That's what I'm talking about, these mixing movements. Now, the propulsive movement is sort of like if you squeeze behind it and keep squeezing behind it, you can push it out. Both of these things are happening at the same time. All right, secretions, you'll see water, electrolytes, organic things like enzymes, bile, salt, mucus. We'll talk about all of these. They're going to be released by hormonal or neural stimulation. There will be hormones floating around that interact with cells that will cause this secretion, or else there will be some sort of nerve that feeds into this area, and when those transmitters are released, it'll interact with those cells and cause these things to be released. I'll explain all this in a minute. I'll give you some examples. Just introducing it right now. As far as a nutrient breakdown, poly means what? Okay, many, multiple, whatever. Polysaccharides, in terms of this right here is going to be in reference to sugar. Okay, so a polysaccharide are carbohydrates, sugars that are made of a whole bunch of different units all stuck together. Okay, now you can start busting those up. Starch and glycogen are actually examples of polysaccharides. Starch, like in potatoes, it's a storage form. Remember I talked about like a roll of toilet paper in the individual squares. We're back here again. This will be like the roll. These are storage forms that can be broken apart into smaller pieces. Okay, so these big things are the polysaccharides. In plants, it would be starch. In animal starch, in us, it would be glycogen, which is slightly different. We can find this in the liver. Do you remember where else we can find glycogen stores in our body? There's two places, big places. One is the liver, because, muscles, that's right, and the muscle tissue, skeletal muscle tissue. Okay, now, these guys can be changed into disaccharides. I'll show you a picture of this. So you can take these polysaccharides, chains of a whole bunch of stuff stuck together, and then it's sort of like a disaccharide would be pulling off like two squares at once. Okay, so because di means two. You'll see that there's polysaccharides, there's disaccharides, and there's monosaccharides, even further breakdown. Okay, glucose is an example of a monosaccharide. Sucrose, which is table sugar, is actually an example of a disaccharide. So there's a little bit of digestion that has to happen when you eat table sugar. It's got to be split one time. I'll show you what I mean here in a second. Proteins are changed into polypeptides. Again, poly means many. Think back to some other biology courses or A&P or whatever. Think about, you talked about amino acids and how you string together amino acids to make proteins. Do you remember what the name of the bond was that holds amino acids together? A peptide bond. So a polypeptide is whenever a protein, a nicely packaged, finished protein, is starting to be digested and it gets torn off into big hunks and long strings of amino acids. Okay? They call polypeptide fragments at that point because there are many peptide bonds that are still held together. And then what will happen through further processing, those individual amino acids will be chopped off and separated. Okay? So we go from polysaccharides to disaccharides to monosaccharides. Then we go from proteins to polypeptides to amino acids. That's the order of breakdown. Now, as far as fats go, fats, if you remember, I don't know if you remember this or not, from when we talked about cell structure and that kind of thing. I don't know which one of these colors are going to work. Let's do black. I told you that a fat was made of sort of a, can't write, this pen's not lining up right. That's supposed to say glycerol, G-L-Y-S, or G-Y-L-C-E-R-O-L. Okay, there's a glycerol group. You see that? Sorry. There's not a good color I can pick, I think, to contrast this very well. Well, I only have black, blue, and red. So, I can try blue. Oh yeah, I guess it is. Okay, there's the glycerol group. Okay, and you've heard of triglycerides, right? Triglycerides are when there's a glycerol molecule with 3 fatty acid chains hanging off of it. They are acid, fatty acids. Okay, so when you start breaking down a lot of fats, some of the byproducts you're going to get are acid molecules, and then acidosis is a concern. With excessive fat breakdown. That's why we've talked about a lot of things, the Atkins diet and all this kind of stuff. Whenever you start breaking down a lot of fat in the body at one time. But anyways, what'll happen is this is sort of like this, a polysaccharide, the large form of sugar. This will be like the large form of a fat. Okay, the difference is in a phospholipid. You remember the things that make up the bilayer in cells. A phospholipid, one of these fatty acid chains is gone, and instead a phosphate group is stuck to it. A phosphate group is a phosphorus atom with 4 oxygen atoms around it. Okay, that's a group, a phosphate group. And it's stuck here at the top because its chemistry is different. So it loses one of these tails, and the phosphate group sticks at the top. Now, as a result of that, that's why it's called a phospholipid. Down here, these fatty acid tails are here, and they are nonpolar. In other words, they don't have any charge associated with them at all, at any level. However, this phosphate group up here is polar. It actually has a negative charge associated with it. So that's why we have hydrophobic and hydrophilic. You remember those terms to describe phospholipids. this would be hydrophilic. Why would that be hydrophilic? Because things with charges are attracted to water. So hydrophilic means it likes water. So this phosphate group that's up here allows it to be attracted to water. This is a hydrophobic end, which allows it to repel from water. It doesn't like to be attracted to water. So that's where the hydrophilic, hydrophobic ends come in. But anyways, if you have fat digestion, what happens is these guys get busted up. You get these fatty acid chains can fall off and get separated, and then our body has to deal with those. And then we've got monoglycerides that form. Generally, the glycerol group doesn't just separate all by itself. Usually, it'll stay attached to one of these chains, and then you'll get a free, you can free however many chains are present. In this case, you would free like a couple of them. But generally, that stays attached to one of them, and that's where the word monoglyceride comes in, down here. Okay? So the end products of fat digestion are going to be monoglycerides and fatty acids that are floating around. Okay? All right, now it says most absorption occurs in the small intestine. That's where the bulk of this activity is going to happen. You're going to see a lot of stuff happens in the stomach. You're going to see there's a complex interaction between the duodenum and the stomach in terms of give and take and feeding back and talking to each other chemically. Once you get past that and get into the small intestine, that's where it's just absorption at that point. And all that absorption, sucking up all that stuff, continues until you get to the large intestine. And the large intestine is just sort of like a drying and storage organ. There's not any absorption that takes place there other than water. Some water can come back in, some water can come back out. We've all experienced both of those situations. So it just depends. All right, so here's a visual of these breakdowns of these complex molecules. Here's a carbohydrate that's a polysaccharide, okay, a big, large storage form of some sort of sugar. What you can use is amylase. Where can you find amylase? Where's the first place the food comes in contact with amylase? Saliva, right. You've got salivary amylase. You've also got pancreatic amylase, which is sort of bad because it's good, obviously, for normal purposes, but we all know that pancreatic cancer and pancreatitis and those kind of things are, anytime you're dealing with the pancreas, you've got some issues that you need to deal with. Pancreatic cancer especially, you know how fast it moves and how dangerous it really is compared to some other types of cancer. When the pancreas starts getting inflamed, if there's tumors growing in it, if somehow these enzymes that we're going to talk about that's associated with the pancreas, because it's more than just insulin that it produces. It actually produces digestive enzymes that are released into the duodenum. If they get backed up, the pancreas can start self-digesting, and it can start ruining a lot of things. It makes digestion not possible. It makes sugar regulation not possible, all of which will have an immediate and very fast and negative impact on the body. So here's amylase, salivary amylase. So as soon as you put it in your body, before you even swallow, you've started the process of digestion. Okay? You're taking amylase and taking these starches especially and busting them up into little units of 2, these disaccharides. So here would be sucrose, for example, is a disaccharide. It's made of 2 monosaccharides stuck together. In this case, you can see what they are. They are glucose and fructose. Okay, those 2 simple sugars. You stick them together, you got sucrose. I'm not going to make you memorize all of those. There's no point, really. But just realize that's all the oses mean sugar. We know that. But realize that some of them, and you have to look them up if you don't know them all, but some of them are simple sugars. Some of them are individuals, like just one of these, which is a monosaccharide, like glucose, that can get into our cells. Some of them, like we eat, like table sugar, cannot go directly into our cells. It's a disaccharide that has to be split. And then fructose, once you get to these monosaccharides over here, and you get to fructose, we can utilize fructose. It's just a small chemical change that has to get it similar enough to what this is to get it inside the cells. So it's not like we've lost that. We can do that. We can convert things and get them in to cellular respiration and use them. We can do that with proteins. We can do it with fats. We can do it with other types of sugars as well. But the key is glucose. That's the reference point. That's what's most important, and that's what everything has to get resembled to in order to be able for it to work. Okay, we'll come back to this in a minute. Anybody lactose intolerant in here? We'll talk about lactose intolerance. Lactose is a disaccharide that you need an enzyme, lactase, right? The aces and the oses, enzyme, sugar. But lactase is actually an enzyme that will break down lactose into glucose and galactose. Okay, and that some people actually don't have this enzyme, or else they either weren't born with it, or chances are over a period of time, which it happens sometimes as we get old, or vice versa even. At some point, the enzyme is no longer functioning properly, whether it's genetic reasons, whether it's physiological reasons, who knows. But when this stops functioning, the person loses the ability to break down that sugar. Now, are you going to starve to death? Of course not, because there's other sugars that you can get. But with lactose being present, there are some things that are living in you that really like lactose. So if by the time that if you have not pulled lactose out of your the stuff that's moving through your gut, through your small intestine, if you've not removed a lot of it already, by the time it gets to your large intestine, it's a buffet for the things living there. Okay, there's still some lactose in there, and there's a whole lot of things like E. coli and everything else in there that likes to eat lactose. So guess what happens when they start eating a lot of lactose and metabolizing lactose? Part of the things that we let go in terms of cellular respiration are gases, carbon dioxide gas. These organisms down here are going to release gas as well, mostly methane, but a lot of other things too, hydrogen sulfide, some other things. That collects in there. It'll either get reabsorbed by the body or else it'll get expelled from our body. So, all right. Proteins up here, we know that individually they're made of amino acids. When we start fragmenting them up, like you see over here, we've got polypeptides. There are some enzymes here you don't have to know the name of. These are some common ones, pepsin and trypsin. They help digest all this stuff that you see. Ultimately, amino acids is the goal over here. I mentioned this a while ago. I told you there was a glycerol group with 3 fatty acid chains to make a full saturated fat here. What will end up happening is this sort of thing right here. Your fatty acids will separate, and you'll keep a monoglyceride, and these are your waste products from fat metabolism. Now what will end up happening is those don't get absorbed the same way that the other things in your body get absorbed, all this other stuff, the vitamins, the minerals, the nutrients. These fats will actually get absorbed by your lymphatic vessels. Think back to your cross-section pictures on your small intestine. Remember they had villi, microvilli, or little villi, and then the cells actually had tiny little villi on those. The purpose of that was to what? Increase what? Anybody? Surface area, right? Area for absorption. What you will see is those guys get busted apart like this. They'll come in through the plasma membrane, not through channels, because they're nonpolar. They can dissolve right through the membrane. They don't have to have channels. They'll get absorbed right through that membrane. They'll come in and they'll go straight into your lymphatic ducts, your lymphatic vessels. And when they're in there, they get actually put back together. They get reassembled as fats, because it's hard to get these things dissolved in water. That's the thing. You guys know that. You've probably cleaned fats and greases before. You know that they don't dissolve very well in water. Well, we're mostly water, so the same thing applies. So we've got to handle these in a different way. And I'll show you sort of a neat thing. Same thing that you do actually when you use dish soap to wash off greases and fats. Our body does the same thing on the inside. We have sort of like our own little dish soap that we use. And we take these things apart, and then when we bring them into towards the inside of these vessels, we'll put them back together, and then we can dump them off into the bloodstream, where you know that the lymphatic system meets the bloodstream with the subclavian, and then they'll dump it back in there, and then it'll get pumped back to the body and take it where it needs to go. So, all right, now this is hydrolysis. Okay, hydro in reference to water. Lysis means to what? To cut something, break something, separate something. To lyse it. So in this case, hydrolysis comes from the fact that it's hydrolysis, using water to split something. Take a look at this picture. Now, you don't have to know a lot about chemistry to just see what happens here. Okay, so make sure you know. Here's a bond that's actually oxygen is holding these two molecules together. These are monosaccharides. Okay, together it's a disaccharide. But right now, what we want to do is break that down. How do you do that? Is chemically you use water. It's called hydrolysis. Water will come in. Notice it's an oxygen with 2 hydrogen atoms. Right? Imagine that coming right over here and wedging itself in here. And look what happens. There's the water, still in blue, H2O. One of the H's hooks on with this oxygen, satisfies the bond that was here, which was all of this. Now it's just one H. Pulls it aside. The OH sticks with this right here, okay, and they separate. You basically have used a water molecule to wedge them apart. That's hydrolysis. The opposite form of this, okay, is what we'll see in plants. It's called dehydration synthesis or condensation synthesis. Why would it be called dehydration synthesis? The synthesis because it's putting together. Dehydration because what do you think happens? Yeah, it pulls out of water. Imagine taking this arrow and flipping it back the other way. You would have to take this, put these together, pull these guys out, and then link this up. Okay? And it's exactly the same thing in reverse. And in reverse, since you're sticking two things together, it's called synthesis. And you're pulling out water to do it, so it's called dehydration synthesis. Here, we call this hydrolysis because you're using water to lyse it. Plants actually make sucrose, like table sugar, sap, right? They do this. They make these simple sugars in their chloroplasts. They stick them together and form disaccharides that then they use to float around all through and feed their tissues. And then we drill holes in it and suck it out. It's like sort of like draining the blood out of a person. It's the same thing, except we're doing it to trees. Okay, the parts of the digestive system you should already know. Mouth, pharynx, esophagus, stomach, small intestine, large intestine, anus. We know that that's the order. We know that it's actually open to the environment in two places, up here and down here. Okay. Now there's a complex interaction here. We're going to start looking up here, how swallowing, what swallowing is, that kind of thing. As we push it down here, the mechanism that blocks off the trachea, okay, which is what? What's that structure that blocks off the trachea? The epiglottis. You swallow, you sort of raise this up, you bring it up, you close that flap and stuff, in theory at least. Doesn't go down the wrong pipe. You'll see that it comes down here, and you can follow the order. Stomach, there's a lot of stuff happens in here. And then, like I said before, there's a complex exchange of communication between the duodenum and the stomach. Okay? All right. Do what? Everything you see here. Yeah. Yeah. Page 582. Yeah. Oh yeah. I mean, this. I guess these are actually technically not an organ since they don't perform a specific function. But as long as you know the entire path from beginning to end, you're going to have included the organs within that. So you'll be fine. Yeah, it's not hard. You should already know this. I mean, that's easy stuff. So. Okay. We do have salivary glands, some exocrine glands, and endocrine glands, as we'll talk about in a minute. And the biliary system, we'll talk about bile. We'll talk about the role of the liver and the gallbladder. What does bile do? Why is it there? How is it controlled? Why is it an issue? That kind of thing. But first, take a look at just the tube in general of the digestive tract. Who thought it would be so complicated? It's not just a tube that squeezes. There's a lot of stuff that goes into it. There's some different layers here. There's innervation. There's circular. There's longitudinal muscle. There's just epithelial cells. We lose a lot of epithelial cells. I mean, there's a lot of stuff that passes through there. Stuff gets scraped off. We've got to be able to regenerate all that. Okay, so working our way from the inside out, we've got the mucosa, which is this lining in here. See this little sort of dotted line out here? Start that as your exterior boundary, and all of this from there in, this would be considered the mucosa. Okay. All right. We've got the inner epithelial layer, which is just this little red stuff in here. This is just the mucosal lining, lubrication, protective barrier, that kind of stuff. Now, inside that, you're actually going to find some exocrine and endocrine cells. So if there's going to be cells that are excreting things into the digestive tract or sensing things here and producing things backwards into the blood, that's all going to happen with these cells in this area right here, these epithelial type cells. Okay. All right. The lamina propria is a middle layer of connective tissue. It's easy to find up here. It's all this yellow stuff. You can see it's sort of just filler space. It's connective. Spaces it out, holds it together. And the muscularis mucosa is called muscularis for a reason. It's got very small amount of muscle associated with it. You can see it right here, sort of that that very outside dotted-looking ring out here. Now, it's not very. That's not what's going to cause your biggest amount of contraction. That's going to be out here, as you can see with this meaty part. But there is a very small amount of it right there. Okay, it's very sparse. All right. Underneath this is the submucosa, sort of that blue area that you see right here. It's most important. The most important aspect of it is the blood and lymph vessels that run through it and the nerve endings that run through it. Okay, it's connective tissue that has large blood and lymph vessels. You can sort of see a little bit. You see these yellow dots here. You don't really see much blood and lymph on this picture, but you do see those little yellow dots. And those are going to be what's called the submucous plexus. A plexus is a gathering of nerves. Okay. Now, what you're going to see and why these are important, obviously, is there's several things that can affect the digestion, just like there is with everything else we've talked about. There are local factors. There's stretch. There's chemical. There's osmolarity. There's acid base. There's a lot of things just inside these tubes themselves that can respond to what's going on. But there's also innervation that comes from a higher source. Okay, that can go down and override things that are happening. So there's got to be innervation close to everything that's going on. All right. The muscularis externa we've got is out here, the big thick part. You can see it. Anyone ever eat any intestine before? No chitlins in here. Okay. The externa is the main smooth layer. It's got there's an inner circular layer. See this? You can see sort of two different textures almost to the muscle that's here. This is a circular layer. That'll just sort of squeeze shut, close things shut. This is a longitudinal layer that would sort of come out at you and go front to back. That could shorten and lengthen the two. Those two work in conjunction with one another. Okay. Now, also, between these two layers, as you can see, sort of right at their boundary, all the way through, you'll see some more of those purple dots, some more nerve innervation. The serosa is the outer connective tissue layer. You can see that out here. Okay. Obviously, these have to be here to control that smooth muscle, right? It's got to receive a nerve impulse. And it's going to be continuous. This serosa will be continuous with the mesentery. Now, think about what that is. Think back to your pig dissection in 102. Remember when you pulled out the gut, especially in the pig, more so than with the human. You pull out the gut, and it was like this big fan of a bunch of connective tissue that had you could spread it all out. It's like a real thin membrane. We have a little bit of that in our body. It was much more evident in the pig. But the serosa is going to be continuous with that. It's going to help hold everything together. Okay? That mesentery is important because there's a lot of, as you will see when we get to the liver, there's a lot of communication that happens through there. There's some blood vessels that go through there. There's some nerves that run through there and that kind of stuff. So. Okay, we know this to be true in terms of how smooth muscle works. It's sort of rhythmic and spontaneous. It varies. It depends on the feedback that it's getting. It's out of our control is the bottom line. It's sort of receiving its own message. It's autonomous. Now, it's just like the heart has pace setter types of cells. You'll find that in our digestive tract we have similar types of cells. The things that, you know what peristalsis is, right? The movement and that kind of thing. Peristalsis and segmentation, the chopping up of this stuff and different segments working at different times. They actually depend on pace setter cells, cells that are in the tract that are initiating this activity and shooting this impulse to start this. So there is sort of some autonomy all there together associated with this. I already mentioned this, the plexi that I referenced a second ago. Those are the enteric. Remember, enterology and enter. Anytime you see that, it's gut related, right? Gastric is stomach specifically. Entero or enterogastric means sort of gut, digestive, all together. So when I say enteric nervous system, you know the nerve leads that are associated with digestion, especially in the gut. So that's what these mean. They are within that, though. When I say the enteric nervous system, that means they respond to themselves. They are local. They're going to process reflexes that are associated with the messages they are receiving from what's happening in the digestive tract. Okay, there are neurons in there that release acetylcholine. Remember that acetylcholine is the main neurotransmitter that's associated with skeletal muscle movement. Okay, so you are going to have here These neurons are going to release acetylcholine. That's what's going to interact with the smooth muscle and cause them to contract. Okay, we do have extrinsic innervation coming from another place. That's not just local. Your autonomic nervous system is going to oversee that, depending on what's going on. Remember that the autonomic nervous system is divided into two things, the parasympathetic and the sympathetic, right? Parasympathetic is going to signal maximum digestive activity. Sympathetic will help decrease this activity. Now, how can you remember that? Remember what sympathetic and parasympathetic are for. Usually when you think of sympathetic, you can think of how does it sympathize with and how does it help you with the fight or flight type of response. Okay? So in the case of sympathetic, we would increase blood pressure, we would increase heart rate. So to increase blood pressure means that we would constrict blood vessels, we would crank up our heart rate, we would dilate bronchial ways, right, to get more air rushing in. Those are all examples of sympathetic effects. Well, it makes sense that in the immediate sense of survival, and when all of this stress effect is happening and sympathetic activity is increasing, what do you want to do about digestion? Well, it doesn't really help you because it is a long process. It takes hours to get this stuff done. So what you actually do is you will find that digestive halts. It stops. It shuts down the blood supply and it shuts down the feeds that are going into digestion to help sympathize with the fight or flight response at that time. Because what you'll see is what we try to do in that type of situation is to most efficiently use our body under those conditions. Take any blood flow where it's being directed and get it away from places it's not needed and get it to places that it is needed, to the brain, to the heart, that kind of stuff. So in a sympathetic, we'll actually decrease digestion activity. So that means parasympathetic, sort of like the opposite, that's when you would have maximum digestion activity. Well, maybe. I'm sure that there's a lot more to it than that, but I don't. Yeah, that's probably just, I don't know, that because sometimes too, I mean, if you're burning up more fuel than what you're using, the first place that goes is sort of in your head. You get lightheaded, and then it's very, very, very easy to become nauseous once you start becoming lightheaded. So I mean, there's the same mechanism, I'm sure, is all tied together in that. All right. We've got endocrine glands. I already mentioned that. There's going to be in the mucosa, so we've got hormones that we'll be talking about. These all same type of receptors are still going to come into play like we talked about before. The chemoreceptors, your mechanoreceptors, your osmoreceptors, all those same types of readings are being taken by the digestive system as well. Okay. All right. This should be pretty simple for you. Hopefully you know what your lips are and your palate, all that kind of stuff. So we're starting now and working our way down. In terms of the organs that we're going to come in contact with, the oral cavity is the entrance. We know what forms the entry, the lips. You should know what your palate, your tongue, and your pharynx is. I don't need to lecture on that. Know that mastication is, of course, chewing. This is where digestion will start because technically you are breaking it down. You're taking it to smaller pieces. And also, you've got saliva in there that's carrying salivary amylase that immediately starts starch digestion, the polysaccharide digestion. And also, saliva will help you dissolve, for taste purposes, it'll help you take those chemicals and dissolve it and spread them out and cover sort of the tongue and let them come in contact with the taste buds, that kind of stuff. Okay, also I've talked about amylase already, but also in terms of saliva is lysozyme. You can find lysozyme in tears as well. But lysozyme is also in there to help destroy any bacteria. It doesn't get all of them, but what you'll see, of course, is as you progressively get farther and farther down, just like with the immune system, we have generic defenses at first that take out the bulk of stuff, and then as things become more complex, we have to increase our complexity in terms of how we fight them. This is sort of just a generic way to get rid of what you can right up front. So we've got some of that. That's self-explanatory. So now, saliva secretion is continuous, and we know that it can be reflexively increased. For example, if you haven't had lunch, and you sit down and you think about the fact that you can get a side Caesar salad with a medium well ribeye. It's 8 ounce, let's say, take it easy, 8 ounce ribeye, medium well, baked potato. And then, or like a barbecue bacon, whatever it is that's at rallies. Oh, that's yeah. You start thinking about that kind of stuff, and then you can start salivating. All right. You know, you guys take psychology, right? You had to take it. You learn about Pavlov's dogs, and how you can condition that salivary reflex. So there is a component there that is not just digestive, but also neural. So there's a tie in there. So that you can reflexively increase them by actually the presence of food or else the thought of. And we'll discuss that a little bit later. But there is a simple unconditioned salivary reflex coordinated by the salivary center in your medulla. But you can also acquire or condition yourself without oral stimulation. Once you learn what things are and you have mental association with what it is that you know that's what it's going to taste like and that kind of thing, you can start that salivary process. So, I think this was a question on, we did a lab already where you did the lemon juice, right? Spit out a bunch of lemon juice and measure the pH changes and the volume changes and that kind of thing. One of the questions, and we hadn't talked about it yet, one of the questions was what division of the autonomic nervous system actually controls the saliva production? It's actually a trick question because both branches of the autonomic nervous system increase the rate of stimulation, increase the rate of saliva production. All right. We know now that once we swallow something, it becomes a bolus. That's the name of the wad of food that we now have. It's not just food anymore. Got to give it a gross name. Now, swallowing. I don't know if you ever thought about how you swallowed or how it works, but whenever you it sort of happens all at once. It's weird to think about, but you have some food in your mouth, you chew it up, and what you do is you use your tongue, even if you don't think about it, sort of reflexive. You use your tongue to push that food back into your pharynx. Sort of all happens at the same time, but it has to happen first that way because once that food starts to contact your pharynx, there are receptors in there that will take that when a food bolus contacts the pharynx, afferent impulses are then sent to the medulla that will trigger the swallowing process. And what that entails is you pushing it back farther, you bringing your throat up, raising up this area so as to lift everything up and close this gap right here. Okay, so obviously as the food's coming down, that'll help push that down as well, but you're raising this up and closing this flap so that it goes back this way and not down this way. Okay? That's the whole swallowing thing. Well, that's what I just said here. So we've got closing of the glottis by the tilting of the epiglottis. It slides back. Your throat rises up. I just said that. We do have what's called the pharyngeal and esophageal sphincter. Big word. Think about where it's at. Pharynx, esophagus. So where the esophagus meets the pharynx, go back here. Here's our esophagus. Here's our pharynx. So we've got this sphincter right here. There's a little round sphincter muscle. It's been cross-sectioned here, but it's there. That is going to prevent air from entering the digestive tract during breathing. That's going to be closed normally as you're not eating. You can't eat and breathe at the same time. We've figured that out, hopefully, at this point. This is interesting. Peristalsis waves. Once you swallow in the esophagus, peristalsis starts, and it starts working its way, worming its way down, sort of doing this kind of stuff. Ring-like. It's sort of like, seriously, it's like putting, like a piece of Play-Doh in a straw and sort of putting your, squeezing behind it and just sort of pushing it forward. You get sort of the same thing. You can get a propulsion towards that direction. A swallowing center initiates a primary peristaltic wave. Now, this is interesting because what starts peristalsis is once you swallow. So when you swallow something and you push it back with your tongue and it comes in contact with your pharynx, the medulla is going to receive that message and it's going to initiate the swallowing response, which will consist of an impulse sent to the area at the top that's going to start that first peristaltic wave. Sometimes, through just haywiring, through something, a misperceived environmental signal, whatever, we get a frog in our throat sometimes. And what that is when this peristalsis has started and they're really, the swallowing reflex had been triggered and that primary wave has been started, but there's really nothing else for it to squeeze, so you don't get a full peristaltic movement. Maybe sometimes you do, but it's when peristalsis has started and there's really no food in there, so you sort of feels weird. They got it kind of shorted a little bit, and that primary wave got kicked in. All right. You got the gastroesophageal sphincter, which is the one down here at the bottom between the stomach and the esophageal tube. That's going to prevent acid reflux from the contents of the stomach to come back up. Obviously, that doesn't work as well in all people. How do they fix the epiglottis? Well, I mean, I don't know that there's, again, I don't really know. I mean, I don't know that there's a surgery that you can do to just replenish everything there, but you can certainly, I guess it would depend on the degree. I don't know. Do you depend upon self-healing? Do you just sort of try to neutralize the stuff that's there? I know there's medication out there that says that actually doesn't just combat the acid, but it actually helps restore the esophageal cells. I'm not sure how that happens. I don't know. I'm assuming that there's I mean, chemically unless if it's a problem that there's a sphincter, if it's a sphincter problem, and there's something they can do to fix that, I don't know. Yeah, find out, I guess, what it is they're doing, and are they putting in something else? I mean, I'm not sure how they would wire that. I don't know how that would work. But okay, but the esophageal secretion that is there is entirely protective. It's not for digestive purposes. It's just to let things slide through and to keep things from harming the cells beneath it. So that's why whenever acid does get up there and starts degrading it, you've got, it burns pretty bad, and that's why its problems long term, like you're talking about, it can sort of eat away at that. Okay. This is the swallowing reflex. There's the bolus in here, the green part. So it's pushed back. Your uvula hangy downy thingy obviously pushes back as you're coming back to keep the food from coming up in your nasal cavity. Some people can do that. You swallow your milk and you can create a compression to increase the pressure up through here and squirt it out back out your nose. But some people do it through their eyes. You seen that? I saw a guy on Letterman do it, but I don't know how he did it. I don't know how it works, but I guess it's possible. I could see certainly how this works, but you'd have to create a lot of pressure to be able to squirt that out your eye. How do you find out you do this? That's the thing.
He could fill water in a balloon with his eyeball, and there was one other thing, and I can't remember what it was, but I was just what?
How do you learn that? And then, like, you see, like, it's kind of weird too, but that's nowhere. Yours is far weirder than this, but like you see pictures of like documentaries and like shows, something like that, but not quite as severe. Just even like things about places from other countries. I saw one about like, yeah, that's exactly what I was in. Like he swallows a snake, and then he guides it, and then it'll come back out, and like the tail's sticking out over here, and the head's sticking out up here, and then he just pulls the whole thing out. Yeah, I don't know. But I mean, who decides that? You know what would be a great idea? Let me stick a snake in my mouth and see if I can get it to come out my nose. I don't understand, because you would have to think about what you have to do. You have to suppress the swallowing reflex, because if you start to swallow, all this rises up, this closes, and the snake's got nowhere to go but down or back out. So I mean, you've got to be able to sort of not ever gag, I suppose. And then how does it know? How does it know to turn around and go? I don't know. It's weird. All right. Okay, the stomach. Largely a storage organ. There's a little bit of digestion. You're going to get some protein breakdown, but nowhere near what's going to happen like in the intestine. So we've got 3 different areas here. Notice you can sort of divide it based upon where it's located if you look. See, here's the esophagus coming in. Here's the cardiac sphincter or the gastroesophageal sphincter. Right across from that, of course, we're all shaped a little bit differently, but right across from that, if you could draw a line straight across, that's the fundus. Typically, that's sort of the area that can be removed or minimized in terms of volume to have the least amount of impact on what's going on here, because, as you can see, just from location, there's far fewer things that are going to be happening up in that area. Down here is the body. This is where most of the stuff's going to take place. Then we've got the antrum. Notice it about right here. If you draw a straight line down, that's about where it starts, and then it cuts straight across. Okay, so. All right. Mostly it stores food, but realize it also secretes these two things. Pepsin and hydrochloric acid. Okay, those are the two big things that you're going to find. And pepsin probably digests what? Look at its name. Pepsin. What do you think it breaks? Peptide bonds. It's going to take apart proteins. Okay. Hydrochloric acid also has the ability to denature proteins. All right. Now, once we get down here, this is not a bolus anymore. Now it's all mixed together in a big sludge we call chyme. Most of this occurs down here in the antrum, sort of all sloshing around. It's kind of neat because you can get up to a 20-fold increase in volume of the stomach. We know that if you ever cut open the one of the pig and opened it up on the inside, you see it's called rugae, those tiny folds. You might not have seen them, but it makes it a very velvety type of feel. When you cut this open, you'll see that there's a whole bunch of little folds so that you can actually, your stomach normally say about the size of your fist, but realize volume-wise it could expand considerably. You can stuff a lot of stuff in there. Once you start getting distended, though, obviously you get some uncomfortable feelings associated with that. But it is possible to store a lot of stuff. Now, think about that in kids, though. That's got to be a higher expansion rate in kids, because how do they eat so much? If you think that their stomach's only about the size of their hand, you think about how little their hands are and how much they eat. You wonder where it goes. There's got to be.
A lot of just eat energy they have, and they burn it off.
That's true, I guess.
Crazy.
All right. All right, now what we're going to talk about is how does our body know when to stop emptying gastric contents? Realize that we don't think about this, but it's very important that our body is able to control how much stuff gets I don't know what the word is, squozen, I don't know, gets squirted. How much stuff gets squirted out of the stomach and gets into the intestine. There's got to be a control there. There's got to be something saying, whoa, 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 I'm backed up here. Keep it for a while. And then there's got to be something else saying, give me some more, give me some more. So there's a balance here. That's the duodenum receiving that and the stomach giving that. So we're going to talk about gastric emptying. Now, what's controlling, what communication is happening that allows the stomach to either empty its contents or keep itself from emptying its contents. Okay? So we know that just peristalsis in itself can carry the chyme out of the stomach and put it into the duodenum. Okay. Now, when the stomach starts to become distended, okay, as it's filling up, you're swallowing more food, it's starting to stretch. There's stretch receptors that say, you know what, there's a lot of food in here now. I need to start increasing the motility. I need to start mixing this stuff up more, get this party rolling. Okay, so that's what's going to happen. So distension will increase gastric motility, and then at that point, as all this is going to tie into a feed from the vagus nerve, it's going to the vagus nerve signal is going to come in, and there's going to be gastrin released. Gastrin is actually released. There's cells in the very bottom of the stomach, down in here, that are, the gastrin is a hormone. It's endocrine. So it doesn't go into the stomach. It actually is secreted out and circulates all around the body and comes back into the feeds that feed the stomach tissue. Obviously, there's blood arterioles that feed the stomach, right? So what you're going to get is gastrin will be produced, and gastrin then feeds back and also helps, once it comes in contact, can now increase motility. So we've got distension. Distension increases motility. Also, gastrin gets released from down here, comes all the way through the blood, comes back into that organ, and when it gets there, it helps increase motility as well. So we've got more movement now. Okay, now in the duodenum, there's a lot happens here. So as soon as the stuff comes out of the stomach and it gets into this area, there are some things that will prevent gastric emptying. There are signals that the duodenum can give back and say, okay, because of this, I need you to stop. I need you to slow down and not give me so much stuff. So what are those signals? Undigested fat. If there's a lot of undigested fat that accumulates in the duodenum, the message that will be sent through a series of biochemical pathways, that's all we need to know at this point. There is a message that is sent that goes and says, slow down, prevent gastric emptying, hold it for a while, let me deal with this undigested fat for a minute. Unneutralized acid. If there's too much stuff coming out of the stomach, chances are there's still hydrochloric acid that was in the stomach. It's going to be mixed in with the food. So what you got to be able to make sure to do is now deal with this acid as it's leaving the stomach. You can't just have all this stomach acid absorbed in your food and let it pass through the digestive system unaccounted for. It'll damage tissue. It'll tear apart your digestive tract. So we've got to have an ability to either neutralize that acid. What can we do to neutralize it? What can we add? Just a generic word I'm looking for here. It's an acid. What can you do to neutralize an acid? Add a what? Right, add a base. What's the most common base that we're going to use in our body? Yes, bicarbonate. Good. Bicarbonate is going to become important again because it'll be produced by the pancreas in this case. Okay, it will help release some bicarbonate in the duodenum to help neutralize any stomach acid. So, if there's unneutralized acid, it's going to slow down. It's going to tell the stomach to slow down, not give me so much, let me have the time to deal with this, help neutralize the situation. If there's an increase in osmolarity, in other words, more solutes, right, than normal, that aren't being dealt with properly. And distension as well. If the duodenum becomes very distended and swollen because it's so stuffed full of stuff, there's mechanoreceptors that will feed back and say, whoa, I'm stuffed here. Let me deal with this. Okay. Now, some more things in general that can prevent gastric emptying. From a neural perspective and a hormonal perspective, there's two chemicals that I will come back to later. Okay, first is secretin. The second one is CCK, which stands for cholecystokinin. Okay, they are enterogastrones. Entero, sort of gut, right? Gastro, in terms of stomach. Ones means they are hormones. So they are hormones that are associated with digestion and produced by cells in this area. Okay, they are actually in the duodenal mucosa. There are cells that make these hormones in the duodenum, and they get released. And if these two guys get released, all this plays in as well. If this happens, you're going to slow down gastric emptying. If these guys happen to get released, you're going to slow down gastric emptying as well. And I'll talk about the mechanism of that in a little bit later. We'll come back to those two hormones. Okay? So just know that these are the two hormones associated with the duodenum, secretin and CCK. All right, now, in conjunction with the sensation of hunger, as you are, if gastric emptying is being inhibited, or if there's, in this, that's because like you think of the duodenum being too full, but realize that if gastric emptying starts to stop, then that's the point when we start to get hungry again. Because when we get into the duodenum and stuff is in there, then digestion is happening. It takes less than an hour, you know, for the stuff to move from up here all the way through here, get into the duodenum, and start through the small intestine. But after everything is sort of processed out of the stomach, and several hours later, as everything is sort of starting to process through the duodenum, we start to get hungry again. So as this gastric emptying is stopping, the sensation of hunger is going to start to increase. Peristaltic contractions are going to begin again to keep this constantly moving. There's never a point where it just, you eat it, you push it all through, and you wait. There's always something going through. And then, you know, the second that you've unloaded this compartment to go into the gut, then you want to refill that compartment. So it's sort of, as that level decreases, it probably never gets completely empty unless you really get growling and you really, you know, you've been a long time since you ate. If you eat when you're hungry, chances are you've not even emptied maybe necessarily everything out of there, and you're just trying to refill and keep it full. Sort of like your gas tank that never empties. You always get a quarter of a tank, you put a little bit more in it. Although it's like $3 a gallon now. So. I was talking about this. Doesn't make any sense. I was talking to my dad the other day. This is completely off topic, but I got to share this with you. It's like he's griping about the gas prices. Every old guy gripes about the gas prices. I was like, okay, Dad, so do something about it. You want to sell your truck? You want to get a new one? Get a hybrid? Get a a dual, something different. No, I ain't buying one of those cars. Well then, shut up about the gas. You know, I mean, come on. Pretty much, yeah. It doesn't matter. No, he's stuck in his ways. All right. Do what? What's that supposed to mean? All right. Peristalsis and hunger. What triggers these things? Number one thing that's going to trigger all of this in terms of controlling digestive feedback, what your brain perceives. Remember that glucose is the only thing that's metabolized by the brain. All your storage forms and all that kind of stuff doesn't matter. That's why whenever you get low on blood sugar, one of the first things that's affected is the brain. The thoughts, the vision, the feeling that you get, the dizziness, all that kind of stuff, cranial. So the reduced amount of glucose being metabolized in the brain is what's going to signal then the need for increased activity, the need for increased eating, food, replenishment, that kind of thing to get this going again. Now, so you can certainly see now, hopefully, that this whole obesity thing and the weight argument that we are currently in terms of the science community and just consumers, health professionals, whatever. I mean, people in general, our culture. Depending on your experience, you can say this is the reason and this is the reason. And a lot of times people like to pick the one that's most convenient for them. But realize that there's no way of knowing. There are certainly a lot of factors that go into the reason that people gain weight and the people that when they do, some people just can't lose it as well and that kind of stuff. Do diet and exercise have a bearing on it? Absolutely. But at the same time, there's some people that could exercise regularly, and there's some people that could diet regularly and do it right and still not be able to lose weight as well as other people. So there's got to be more to it than that, although that is definitely important. Some people genetically are just predisposed to being bigger people or smaller people. The more we learn about this, you see that there are a lot of factors that control digestion. There are a lot of factors at the hormonal level, the neural level, that we'll talk about leptin a little bit later. When we get into especially when we get into a hormone chapter, we'll talk more about this. But there's a lot of feeds that could go wrong. There's a lot of enzymes that could go wrong. There's a lot of things that are secreted that could go wrong. What about this? I mean, think about a person who realized that there has to be receptors. There's got to be a complex pathway of neurological response and neurotransmitters that are manufactured from the genetic code in people's cells. There has to be something here that is sensing the level of glucose. Well, what if this person's doesn't work as well? What if they're constantly feeling as if their receptors are saying they need more glucose, and they have nothing to do about it, so they have the desire to eat more. Can you physically suppress that? Can you decide not to? Sure, but that's easier said than done, right? So there's a lot that goes into it. I just wanted to point that out. So, but that's not an excuse either. I'm just saying there's a lot that goes into it. Emotions can certainly influence gastric motility, especially in reverse, I say, or exceptionally fast forward. Either way, but. Talking about vomiting here. The stomach does not, however, participate in vomiting. Your stomach doesn't squeeze to project its contents out. Your abdominal muscles squeeze your respiratory muscles more than anything else. This is why you can't breathe either when you're vomiting. But you can feel that. It's right here, especially if you've ever had a good, nice vomit, to where your stomach hurts when you're done. I've had those before, right? Your stomach hurts, and the next day it's like, oh. But actually that's those respiratory muscles that usually don't get called on to force that expiratory reserve that's out of your body, bring them back from the respiratory chapter. Those muscles that they get called on now, and they help compress all of this. It ends up compressing the stomach, and it helps propel that outward. You've got tactile stimulation, irritation or distension. You could put some things in there that you have an adverse effect to, or adverse response to. It could irritate your stomach lining. It could, if you have distension, if you eat too much. I've been guilty of that. It's a great country to live in whenever you can eat so much that you get sick. I've been there before, especially when it comes summertime and you eat melons. I don't know if anybody likes melons or not, but I have a problem, a serious problem with that. I sit down and I have cantaloupe, watermelon, and I'll actually eat until I get sick. I can't put it down. It's the only thing really that gets me, though. So, anyways, elevated intracranial pressure. Anybody who's had a headache can even know sometimes you can have headaches so bad that it makes you throw up. I'm sure you can speak from personal experience with this. But there's also just chemical agents. You inhale some noxious fumes, whatever. Psychogenic factors. I hope this hasn't happened, but we all lead busy lives, and a lot of you have a lot of things going on besides school right now, and stress becomes an issue. We know that I can speak from experience as well that you can get so stressed out that you just throw up. I It makes you physically sick. These are psychogenic factors. Now, that doesn't mean that it's all in your head. That's not what I'm saying. I'm saying it starts here. And then what trickles down because of that is a set of physiological events that can be associated then with all the other stress factors that you are experiencing, that you feel. All right. And obviously, severe vomiting can lead to dehydration and circulatory problems. Dehydration is obvious. Circulatory problems, why? How could it affect your circulatory system? There you go. If you get dehydrated, it's because you're losing water. If you're losing water, how does that apply to blood vessels? Yeah, you're going to lose blood pressure, right? You lose volume, like you say, and because of that, you're going to lose blood pressure. So, I mean, it's more than just becoming dehydrated. You've got, hypotension could set in. In addition to that, our acid-base balances could get screwed up, right? Think back to acid-base chapter. We were projecting a lot of acid out of the body. Alkalosis could come back in because there's too much base now. The levels were working in conjunction with one another, and then you get rid of all the acid, so now you've got all this base left over. I mean, there's a lot of issues here. So, not to mention that, but people who induce vomiting in themselves on a regular basis. We've got to think about, esophageal damage, certainly irreversible even, esophageal damage. A lot of mouth damage. The acid can eat away at the mouth, the teeth especially. So there's a lot of things that can go wrong with that. Okay, as far as the digestive juice, this is some pretty potent stuff. Your pH around 2 maybe. This is pretty not concentrated, but pretty strong acid that's in your stomach. We know this because it digests things. Okay. All right, we've got a couple of different types of cells. We've got what's called parietal cells and chief cells. And they're in these pits that when you have the stomach and you look at the tissue, you'll see these little pits all over the stomach, and they dip down in, and these gastric pits down here are where these cells are that secrete what's ended up going to be pepsin and hydrochloric acid that's going to come out here and be in this area. So if I look at parietal cells, it says parietal cells secrete hydrochloric acid and the intrinsic factor. Here's a parietal cell right here. I'll show you how this works with an animation. But if you take this parietal cell, it actually goes through a whole transport system that kicks out hydrogen ions and chloride ions that we call hydrochloric acid. Let me get rid of this. Hang on a minute. Okay. So that's there. Also, we've got some cells, some chief cells that are going to release something called pepsin, or pepsinogen actually. And then when pepsinogen is released out here, it can now interact with the hydrochloric acid that's present to produce pepsin. So now we've got hydrochloric acid, we've got pepsin. Those two together will start breaking down proteins. Okay. All right, let me take a look at, I'll talk to you about intrinsic factor. Intrinsic factor is, what it does is it helps us absorb vitamin B12. We need vitamin B12 for some of the biochemical pathways that exist in our body. And without this intrinsic factor, without our stomach secreting this chemical into the gastric lumen, we can't absorb it correctly. So otherwise we wouldn't get it. All right, let me look at this here. It's going to work. There we go. I don't think there's any sound to it. What? Just get this out of the way. Okay, so take a look. It's showing you the parietal cells. It's nothing fancy here, but we're in a gastric pit here. Back to these pumps. These things that grab on one end, flip it around, take it where it needs to go. Histamine can come in to do this. Gastrin, which is a hormone I mentioned a while ago, they can come in and trigger these cells to help take hydrogen from what's in here and pump it out through this way. Now, when they start pumping that out, you start filling up that gastric lumen with hydrochloric acid. Okay. Nothing fancy there, but just a visual. All right. Like I said, the low pH, which means the increased level of acid that's in the stomach, that will help convert the pepsinogen that's been released by the chief cells into active pepsin. And then those two things together, pepsin and hydrochloric acid, can start to help digest these protein fragments, okay, and bust them up a little bit more. So as far as the functions of hydrochloric acid, we've got pepsin formation, which is breaking down pepsinogen into pepsin. We've got the breakdown of connective tissue. So think about eating meat. You know, that clear stringy stuff that's on the meat and all that kind of stuff, that has to all be broken down. And even at a smaller level, there's connective tissue holding everything together that we eat. We've got to break down some of that connective tissue. The acid will help do that. And the muscle fibers in the meat that we eat. It'll help denature proteins. It'll help take them as they're all wadded up, sort of turn them out and separate them and unwind them. And, of course, killing any microbes that may be present at that time. Okay. So the mucus lining then on the surface of the gastric mucosa, it's all protective. There's some there's some research going on down the road at ISU, but there's also some neat information out there. They just actually didn't always know about the fact that ulcers, of course, an ulcer is just a breach in this mucosal lining, right? And whenever this acid can leak into that ulcer, obviously it can burn because that protective barrier is gone now, and you get an acid reaction. We now know, of course, that it can be bacterial in nature. Sometimes Helicobacter pylori, which are a species of bacteria that can get in there and actually, very rare because most bacteria are killed by that high acid environment, they can get in there, dive beneath sort of like that mucosal layer and protect themselves and colonize and actually eat away at that layer and expose sites and breaches in that mucosal layer that allow the acid to react with that area. So depending on the type of ulcer, you know, stress-induced ulcers, you know, what does that mean anymore? Because it could be that your stress response now has decreased your immune system. Now, actually, it led to an infection, and that infection is what caused the ulcer, right? Bacterial. Or it could be for a variety of other reasons. So it's hard to tell now. It depends on what's going on with you in terms of how that treatment would work. So, all right.